Welcome back. This is lecture 2. We're starting on page 671, talking about the pancreas now. Notice on your lecture outline, the first thing that you'll want to do, not necessarily right now, but eventually, is just go back and review figure 1723, which we've talked about in lecture. Let's jump down now then on your lecture outline to the section labeled pancreatic juice. And so just as gastric juice in the stomach, from the stomach, contains various substances that are involved in digestion, so also now we have pancreatic juice, this mixture of substances produced by the pancreas, which again help with digestion. So to your outline then, this pancreatic juice contains the enzymes amylase, which breaks down starch and glycogen into disaccharides. The pancreatic juice contains lipase, which you'll recall breaks down fats into fatty acids and glycerol into their component building blocks. Pancreatic juice also then contains enzymes called trypsin and chymotrypsin. And note then that these are breaking down proteins or partially digested proteins into peptides, into smaller pieces. Carboxypeptidase, another enzyme, which breaks down the peptides, these small pieces, these small chunks of what were originally very large proteins, and breaking them down now into individual amino acids. And then finally, pancreatic juice also contains various nucleases, from the name then, enzymes that break down nucleic acids. And recall from ANP1, nucleic acids are DNA and RNA. And these nucleases then are breaking down these nucleic acids into their building blocks, into these structures called nucleotides. So, regulation of pancreatic secretion, figure 1724 on page 672. Take just a minute, since this is a new diagram now, this is a new figure, and just notice the organs that we're going to be looking at here. Notice the direction in which this cycle of events occurs. And so starting off with number 1, and then following the arrows around to number 4, and then finally we go to number 5. We begin then with acidic chyme entering the duodenum from the stomach. And notice here we're specifying acidic chyme that is more higher, let's say, in hydrogen ion concentration, chyme which is somewhat acidic. And this chyme then comes from the stomach, enters the duodenum, and this stimulates the duodenum to secrete a hormone called secretin. So again, remember when we're talking about these various chemical compounds involved with digestion, we're talking in some cases about hormones, and in other cases about digestive enzymes. And again then, secretin is a hormone. It is then, of course, secreted into the bloodstream, as is the case with all hormones. When the secretin reaches the pancreas, it stimulates the secretion of this pancreatic juice that we were just talking about, and specifically, it stimulates pancreatic juice release with high bicarbonate ion concentration. And bicarbonate is a basic compound, is a basic solution, basic in terms of acids and bases. Following along then, when the bicarbonate ions move from the pancreatic duct, and so take just a minute now and follow that pancreatic duct in the figure all the way down to the duodenum. And when the bicarbonate ions then move into the duodenum, they neutralize the acidic chyme. Down below then, notice now we're talking about chyme containing proteins and fats entering the duodenum versus chyme which is simply acidic, as we were just talking about, chyme which doesn't really contain much in the way of nutrients. And in the case then of chyme that does contain nutrients, that does contain proteins, and does contain fats, when this type of chyme enters the duodenum, it stimulates the small intestine to release the hormone cholecystokinin, CCK. And this now is the second time that we're mentioning CCK with respect to what it does in terms of digestion. There's not a figure in your textbook that's specific to the release of CCK, but you can go back to figure 17.24 and follow that pathway. And the first difference then is that the intestinal mucosa, step number 2, is now going to be releasing CCK rather than secretin. In the next step then, number 3, the secretin in this case stimulates the pancreas to secrete not bicarbonate ions, but rather pancreatic juice rich in digestive enzymes. And when the enzymes then move from the pancreatic duct into the duodenum, they digest the proteins and fats and other nutrient molecules that stimulated this whole process in the first place. Let's go ahead and get into talking about the structure of the liver now. And so 17.7. And notice we're going to be looking at figures 17, 26, and 27, and 28 on page 674 and 675. So to begin with, on page 674, notice in 1727A, we're seeing a structure called a hepatic lobule. You'll remember these structures from laboratory. You'll recall also then that these hepatic lobules contain canals, contain pathways called hepatic sinusoids. These sinusoids carry blood, and they carry blood then to a central vein. So take just a minute, look at 1727B now, the lower right-hand portion of the page. And let's go ahead now and talk about several different blood vessels. Notice there are no fill-ins here for you guys, so you will be taking some notes at this point. And so we're talking first of all then about a vein called the hepatic portal vein. Take a minute and notice on 1727B, again lower right on page 674, we're seeing one of the branches of this hepatic portal vein. And you should jot down in your notes then that the hepatic portal vein carries absorbed nutrients from the small intestine. Notice also, back to our lecture outline, we have hepatic arteries. Go back to your figure 1727B, lower right-hand part of the page again, and locate now one of these branches of a hepatic artery. And these hepatic arteries then supply oxygenated blood to the liver. And of course, the liver is going to need this oxygenated blood to do cellular respiration to produce energy for the cells of the liver. And then finally, notice back to your lecture outline, there's a vein called a hepatic vein, which is not diagrammed anywhere in this area of your textbook. What you can jot down here in your notes is that the central veins feed into the hepatic vein, and the hepatic vein then carries deoxygenated blood away from the liver. Let's take a look now on page 675 at figure 1728. take a minute, orient yourself again with what we've just talked about in terms of oxygenated blood flowing from branches of the hepatic artery. Blood also deoxygenated, by the way, for the most part, flowing from branches of the hepatic portal vein. Again, follow the arrows to the central vein, upper right-hand corner. And now take a look and notice that this figure also shows structures called bile ductules. So essentially small ducts, small tubes. Locate then one of these bile ductules and notice that the arrow shows that the flow of bile through these ductules and to the bile duct. Notice that the flow of bile then is moving in an opposite direction from the flow of blood through the lobule. In terms of liver functions, notice there's a table, table 17.7, on page 675. And looking at that table, you can see various general functions of the liver are shown here. And then off to the right, for each of these general functions, one or more specific functions are being presented. So what I'd like you to do, and again, I don't want to take the time right now to have you jot down a bunch of information in your notes, but what I would like you to do at your leisure is from table 17.7, be familiar with the 7 general functions of the liver, so carbohydrate metabolism, so on, all the way down to secretion. And then for each of these, I'd also like you to be familiar with at least one specific function within that category, within that general function. I'm going to close the lecture here, and we'll come back and talk about these functions of the liver beginning with lecture 3.